So, this is about as detailed into the molecular biology as I'll go, but starting to get back to the metabolic engineering perspective, I wanted to talk about how this might practically impact how you might do some engineering. So let's use the example of coumarate production. Coumarate is an aromatic carboxylic acid. It's used in a variety of industrial syntheses and kind of a building block for a lot of natural products. It's one step downstream of tyrosine. So, for a pathway like this, a lot of the same operations that you would perform to try to increase flux to tyrosine are essentially what you would do, plus heterologous expression here of what is probably a tyrosine ammonia lyase to get to coumarate. So I have a question for all of you as you look at this example from the literature, which I believe is this pathway to coumarate developed in Saccharomyces cerevisiae. You can see, for example, the knockout symbol here applied to arrow 10. And yeast in its aromatic amino acid biosynthesis pathway has a number of enzymes that are multifunctional. And this is also important for different reasons. In this case, it's convenient. Maybe if you want to prevent all of these reactions at the same time, you can just do a simple knockout. But if you had a multifunctional enzyme that led you past, say, a branch point, and you wanted to eliminate some but not all of the steps, that's another case where a knockout wouldn't be appropriate. So my question for the audience here is, in addition to this knockout of arrow 10, what do you see represented in as a strategy that the authors took here that has to do with the topic that we've been focusing on, predominantly this sort of metabolite-based regulation?
It looks like they mutated arrow 4, turning I don't remember what K and L are. I'm blanking. I think it's lysine and leucine.
That's
Lysine and
right.
glucine.
Yeah.
I'm not positive what that did, but they did mutate it, as well as arrow 7.
Yeah, good observation. So if you had to guess, so you're not positive what it did, but if you had to guess, what do you think it might do? What would you want it to do?
I imagine, at least for arrow 7, they wanted to get rid of the substrate inhibition because if there was tyrosine buildup, that would stop the pathway towards coumarate. I guess same thing with arrow 4.
That's right. So that's very good. The only minor caveat to your answer is it's not substrate inhibition. It would be product inhibition in this case. Or typically product, I guess, for an enzyme implies the immediate product. But yeah, a downstream inhibition of some kind. So that's exactly right. I mean, if you relied on knockouts alone, you wouldn't be able to address that precisely because of this post-translational form of regulation. And if you think about some of the other tools that we've discussed, like metabolic flux analysis, modeling approaches, they would say, hey, you need to increase flux through this pathway. But they're not going to point out, very few, if any, of those models would know that you would tell you first that you need a that you can't actually just do a simple upregulation, that you need to do a mutation, and they wouldn't know what the mutation is. So this is an area then where I believe these enzymes shown in blue are actually overexpressed. And so the TAL protein is a heterologous one. Tyr-C, arrow 7, this variant, and arrow 4 are probably ones that, they are endogenous variants, probably ones that were actually found from evolution experiments or mutational experiments. And this is, I think, one of the things I also want to emphasize here is this idea of where engineering and science intersect really nicely, because it's very unlikely that it was engineers who found these mutations to begin with. When you go back in the literature, it's actually molecular biologists saying like, hey, we found this strange form of bacteria that's like overproducing tyrosine. How's it doing that? And we isolated these mutants. Or we found that if we added some compound, it made cells really sick. But as we evolved them or irradiated them with UV, we found a few mutants that were no longer inhibited. And that's usually the characterization that follows that, ends up revealing really useful tools for engineers to eventually use. So I'll just cover another example that's very related, and then pause for questions and move into the topic of what should be today's lecture. So this is another example, just to, related to your homework also, to show you that this is more than just an aromatic amino acids. Still an amino acid biosynthesis pathway. It's that for methionine, also very complex. Methionine has a sulfur atom within it and comes from a cysteine precursor, which has this thiol. And methionine also requires a methyl transfer event. And so it may be, if it's not clear here, it might be in the next slide, that some of these are energetically expensive. You can see the pathway starting from aspartate, and immediately there's a need for some ATP and some NADPH, and then again, and then, right, you have the cysteine and so forth. So in this pathway, there is more regulation than what is just shown here. But this was another example of some metabolic engineering work done, this out of Japan, showing first some of the negative regulation steps that are built into this pathway. And it's interesting, some of these look like they're off pathway. I mean, you've got this core aspartate going to methionine, and then you have cysteine shown here. And actually the cysteine biosynthesis pathway would be a little bit more interesting to look at, has its own regulation. But there is at least this feedback from S-adenosyl methionine. So methionine is complicated, more complicated in some sense than some other amino acids, because SAM is the cell's methyl donor. And so although every amino acid essentially has a downstream sink in the form of, it's going to go into a protein at the very least, if not some other structures. In methionine, there's even additional demands. So for example, any of the DNA methylation that might be happening in your organism is coming from SAM, and therefore from methionine. And so anyway, there was some knockouts shown here downstream of methionine, and effectively you would think that might eliminate, at least as illustrated here, all of your SAM, but there's probably something more complex here occurring, such that there's still some SAM available, which I'm actually not sure I would think is essential to the cell. In either case, there's yet another knockout performed here to prevent some kind of negative feedback there. And so anyway, the details of this pathway are actually, well, this particular pathway, not super familiar to me, but in your homework, you'll see a little bit more on E. coli's biosynthesis of methionine and SAM. And that was something that I've looked into before. Okay, so just to wrap up this idea of native gene regulation, as it's getting us closer towards thinking about how we would engineer things. So nature's come up with all these different ways to regulate genes and proteins. So if you want to overproduce a metabolite, you probably have to do more than just knockouts predicted by MFA. And you can actually start to design constructs. I mean, maybe just like in a chemical plant, you want some kind of control. Like, it's not just nature that wants homeostasis. If you want to prevent a runaway reaction in a normal chemical plant, you try to design negative feedback sometimes into your process. You don't see that as much in metabolic engineering. A lot of times you just see, let's overproduce this metabolite in batch. But what you might find a lot of are examples where you want to do that, where you turn certain things on, and you have other pathways that you want. I mean, really, when you think about it, if you want to overproduce some metabolite of interest so highly, you still need to make proteins. And so in a way, you actually need to maintain a fine-tuned regulation and some kind of homeostasis of all your amino acids, because they are needed, and to then make the enzymes that you use. And finally, if you are overexpressing constructs, it's not just as simple as overexpressing the native gene. And even as you express foreign genes, you've got to think about possible areas for regulation. So I'll pause for questions here, mostly related to gene regulation and aromatic amino acid biosynthesis or methionine biosynthesis.